How does a smooth muscle know when to contract or relax? Let's explore this in Learner's Vision Pharmacology, Pharmacodynamics Part 3, Ligands and Receptors. In the previous video, we saw that smooth muscle contracts when MLCK is activated or MLCP is inhibited and relaxes when MLCK is inhibited or MLCP is activated. To understand this further, let's take the example of vascular smooth muscle during exercise. When we work out, our skeletal muscles need more oxygen and glucose. To meet this increased demand, more blood has to reach these muscles. But the body has only so much blood available at any given time. So how is this possible? An important mechanism is redistribution. That is, temporarily redirecting blood from less active regions like gut and skin to the skeletal muscle. For this to happen, the arterioles supplying these regions must constrict, while those supplying the skeletal muscles must dilate. In other words, vascular smooth muscle in different regions must respond differently. This brings us back to our original question. How does the vascular smooth muscle in skeletal muscle know it should relax, while smooth muscle elsewhere knows it should contract? The simplest method is to have messengers, molecules that are released when the body is exercising. So where do these messengers come from? When an individual anticipates exercise, the brain sends a message through the sympathetic nervous system to prepare the body. The primary messenger of the sympathetic system is the neurotransmitter norepinephrine. And the effect of norepinephrine is vascular smooth muscle contraction, leading to constriction of arterioles supplying the skin, gut, etc. But the sympathetic nerve endings also innervate the vascular smooth muscle in skeletal muscles. So instead of dilating, wouldn't the skeletal muscle vasculature constrict as well? No, here is why. Skeletal muscles contract by using energy from ATP. We know ATP breakdown releases adenosine. Since this happens in the exercising muscles, it makes perfect sense for adenosine to act as a local messenger. Can you guess what the message would be? For the smooth muscle to relax so that arterioles dilate and blood flow increases. Thus, in skeletal muscles, adenosine overrides the vasoconstricting message of norepinephrine. But there is an issue here. Norepinephrine and adenosine are polar molecules and they do not cross the cell membrane. Then, how does this message reach the enzymes which are located within the cell? Can you guess? Here is how it happens. There are molecules on the surface of the cell to receive such messengers. These molecules that receive these messengers are called receptors. So we have norepinephrine receptors and adenosine receptors. The messenger molecules bind to these receptors. To bind in Latin is ligare, and from this arises the word ligand or ligand, the term used to describe any molecule that binds to the receptor. Now the question is, how does the binding of a ligand to the receptor on the cell surface lead to an effect on the enzymes located within the cell? For this, we have to understand the basics of ligand-receptor interactions and downstream pathways. We will begin that discussion in the next video. Let's summarize. To meet a physiological need, the body often releases messenger molecules. Those that bind to the receptors on the target cells are called ligands. Ligand receptor binding initiates downstream pathways within the cell. This results in cellular effects and ultimately helps in fulfilling the body's requirement. These are the two important terms in this discussion. Here are some good to know facts. This is a must know fact.